This video, I'm going to explain a polysaccharide known as amylose, which is a component of starch. It is also known as a complex carbohydrate. The key function of amylose is energy storage in plants known as starch, and it is the main carbohydrate in human diet and are found in rice, sago, and wheat, etc. It is also food reserve for plants by a process known as photosynthesis. So let's take a look at how amylose is formed. So firstly, I have a glucose molecule here known as alpha-D-glucopyranose. It is a monosaccharide or monomer of amylose, and it is in a cyclic form. What is important to know about this glucose is that it contains a 6-carbon. This monomer is a smaller unit which can create larger molecules like amylose. So let's take a closer look here. The C represents a carbon atom, and we can label these carbon atoms of glucose with number 1, 2, 3, 4, 5, and 6 in its specific order. At the CH2OH group of the chiral carbon determines the deconfiguration of this glucose. So in carbon 1, this is the anomeric carbon. The OH group on the anomeric carbon is below the plane. So now we have learned that an alpha-D glucose has a 6-carbon atom. This type of glucose is named alpha because it has an alpha configuration. Alpha carbohydrates is where the hydroxyl group of the OH group of carbon number 1 pointing in the opposite direction of the carbon number 6. So these are opposites to each other. So now, how the linkages are formed? It is formed when a glucose molecule, this glucose molecule and another glucose molecule can form a bond with each other. It is linked together by an alpha 1 to 4 glycosidic bond. Because carbon number 1 of one glucose and carbon number 4 of another glucose are involved in the linkage process. Now, the process of linking monosaccharides with one another is called a condensation reaction or a dehydration synthesis. Here we have OH group and another OH group, which is known as H2O or water. And water is released in the condensation reaction. This process requires energy. Now, a glycosidic bond forms and holds 2 of these monosaccharides together by an alpha 1 to 4 glycosidic bond. So each time an alpha-D-glucopyranose is added, it's a condensation reaction adding on another unit of the alpha-D-glucose every time. So we basically form a disaccharide first, and we add a couple of more of these alpha-D-glucopyranose that exceeds 20 with more than 20 bonds. This carbohydrate is referred to as a polysaccharide. So now we can say amylose is a polysaccharide, and it is a homosaccharide because it contains only one monosaccharide linked together in an unbranched chain of thousands of subunits. It is held by a hydrogen bond between the CH2O groups. This is what pulls and twists the chain. So this is amylose in its helical structure. So the hydrogen bonds between this helical structure is what pulls and twists the chains, and this allows it to store in large number of molecules in very small spaces. So amylose is insoluble in water. The question is why? The short answer, it is relatively nonpolar. This hydrogen bond that forms the internal network provides a barrier to water penetration inside the granules that are therefore insoluble in water. So this is my demonstration here. So we can say that this is amylose, and this is our water molecule that is polar, and this is non-polar. So as you can see, it is insoluble. Amylose is insoluble in water. So in conclusion, amylose is a polysaccharide consists of a linear structure of alpha-D glucomonomers linked together through an alpha-1 to 4 glycosidic linkage. It is helical in structure and possesses the H atom, but is hydrophobic in nature. Thank you.